Okay, welcome to week 7th. So, in week 7, basically, we are going to do one lab. This lab you will do at home, and this lab has been provided to you in the Carolina kit. So, and also in this lab, you are going to do a discussion forum. I have posted on Mercer Blackboard one research paper. So, just try your best to understand that paper. I know it is difficult paper. Just try to understand what is the main idea of that paper, what those people are doing in their research, and how bioinformatic tools have helped those researchers in their research. And that is what is the topic of that discussion forum, that how bioinformatics tools they have helped in this research. Okay, so basically you have to go and you have to write your comments in the discussion forum. So that is the another activity for this week. So discussion forum is not going to be graded, but it is required. Everybody needs to participate. And the second thing is the lab. This is lab number 4. So this is from the Carolina kit. So we have done 7 labs in this course. So 3 were computer labs, and 4 are the virtual and the labs which are going to give you hands-on experience. So basically in this lab, what you're going to do, in this lab you are going to do DNA electrophoresis. So basically in this kit you have been provided for your experiment number 2, which is our lab number 4. This all material has been provided to you, and with the help of all this material you will be able to run the DNA gel. So this basically lab has 2 parts. There is and also there is a worksheet for this lab which I have posted on Mercer Blackboard in week number 7 folder. You have to, and it is worth 15 points, you have to answer some questions in that worksheet. So I'll be explaining you as we are going along with this lab. So basically in this lab, first of all, you are going to do an activity which is basically on the basis of your understanding, which I'm going to explain you. So you will be doing some restriction enzyme digestions that you will be basically working on a piece of paper, and then you are going to note down your results, and you can insert your answers in the worksheet. And second part is you are going to run a DNA gel. So this is all the material. So all this material should be there in your kit. So this is also called DNA electrophoresis. So the first activity is linked to the second activity. So second activity is running the DNA gel. So the first activity tells you why do we need to run the DNA gel. There are various situations where we need to run the DNA gel. So I will be discussing with you one situation where we need to run the DNA gel. So the kit which has been provided to you, it does not have actual DNA samples, but it does have some dyes, so which can just substitute the DNA samples, the real DNA samples, but it can give you an idea how DNA electrophoresis is done. Okay, so I'm going to talk to you about a situation like if we have a DNA sample from we have 2 DNA samples. One is from a normal person, another DNA is from a person who is suffering from sickle cell anemia. Okay, now the people who suffer from sickle cell anemia, their red blood cells, they are sickle shaped, and their oxygen carrying capacity is decreased as compared to the red blood cells in the normal people. So why do they have different red blood cells? Because they have a mutation in their DNA. So whatever I'm saying, everything is there here. You can go over this also. So they have a mutation in their DNA. Okay, and this mutation, so there's a point mutation. point mutation means it's a mutation in just one nucleotide, or basically the nitrogenous base. The point mutation is a simple switch of one adenine nucleotide to a thymine, and this single change has a significant results. This mutation changes a glutamic acid to valine, altering the shape of hemoglobin in such a way that blood cells become curved and they misshape. So basically, there is a point mutation, there is a change in one nucleotide, and that change in one nucleotide changes the amino acid, and that changes the protein. Okay, so that changes the shape of the hemoglobin, which is a protein, and that eventually changes the shape of the red blood cells. And these people suffer from a lot of problem. So now if we want to detect, like how to tell that this DNA is from a sickle cell anemia patient, this DNA is from a normal person. So basically what you need to do, you need to go and check for that mutation in the DNA. So you need to check to find, to state that this DNA is from sickle cell anemia patient and this is from normal person. To know that, you need to go and look for the mutation. But now, how to check the mutation? So to check the mutation, first of all, basically there is a specific area in the DNA where that mutation is. Okay, so basically, if you want to check that area, you want to check the mutation in that area, you need to first of all amplify that region. So amplify that region means you need lot of that region, because otherwise it is going to be so small you cannot analyze and you cannot detect a mutation. For that, a technique called PCR or polymerase chain reaction is used. So PCR is a lab-based technique by which we can amplify the DNA. We can make multiple copies of DNA. So it's done in the laboratory. So and it works on the same principle as DNA replication. So we have already talked about how does DNA makes its copy. It's a semi-conservative mode of replication. The two strands, they're going to separate, and then each strand is going to act as a parent strand, and then there will be formation of a daughter strand which is complementary to the parent strand. So this is the way how DNA replicates inside your body, in your cells. The same principle has been used, and it is used in the lab, and by which large amount of DNA can be formed. So here this is all it has to be like it's done in a tube, in a test tube, and there is a machine on which those tubes are placed. Like in the tubes, you provide all the ingredients, you provide the template DNA, you provide primers, you provide DNA polymerase, nucleotides. Now what is a primer? Basically, the enzyme which is involved in the replication of DNA is called DNA polymerase. DNA polymerase cannot itself initiate the process of replication. It needs a short stretch of DNA. Okay, inside our body, inside our cells, the primer is RNA, but in this technique the primer is DNA. So DNA polymerase needs a short stretch of DNA on which it can elongate the new strand. Okay, so DNA polymerase cannot initiate this process, but it can elongate, and that is what is the role of primer. It gets the 3' free end of the primer, and DNA polymerase adds the nucleotides at the free 3' end of the primer. So basically this is a technique in which all the ingredients they are added in the tube, and this machine in which the tubes are placed, it is connected with a computer, and there is a change of temperatures which are regulated by the computer program. Basically, first of all, the first step is denaturation. The denaturation is when the two strands of DNA, they are going to separate, because at this point the temperature is very high, which makes the two strands separate. Then second step is called annealing, and this is the step when the DNA to the template DNA, and this temperature has to be it has to be calculated on the basis of the primers you're using. And then last is extension. So once the primers they have attached, then is the extension. I think it is usually around 72. Then the once the primers have attached, DNA polymerase they can then elongate. So this is the way how DNA can be amplified. The multiple copies of DNA can be made in the lab. So the region where the mutation we suspect, that region should be amplified, because if it is less we cannot detect the mutation. So we will amplify that region by using this technique called PCR. And you can see here, this is all this technique is showing. This is your double-stranded DNA. So the two strands will separate. These are the primers. One primer attaches here, another primer attaches here, and so first of all they separate, then primers attach. That step is annealing, then it's extension, and this is the way the DNA is going to be amplified. Okay, so once the DNA has been amplified, which means you have a good amount of DNA now, what is the next step to detect the mutation? The next step to detect mutation is you are going to use enzymes called restriction enzymes. Now they are enzymes, which means they are protein in nature. Why they are called restriction enzymes and what is the function? Restriction enzymes are enzymes, they are proteins which has the capability of cutting the DNA at specific places, and by cutting the DNA they act as scissors. So like a scissor can cut, anything you want to cut, you can use a scissor and it will cut. So restriction enzymes, they are enzymes, they are proteins which can cut DNA, okay, which can cut DNA. But where they cut, these enzymes, they are very specific, and there are different kinds of enzymes. Okay, so if you are working on a restriction enzyme, first of all you need to go and check at which specific sequence it is going to cut. So there is a special recognition sequence for every restriction enzyme, and there is definitely a way how that enzyme cuts. I'm going to show you one table where everything is written there, and also these are kind of questions are there in your worksheet also. So if you know about a restriction enzyme, some information has been given to you, you have to answer some questions. So I'm going to go over with you guys. So why they called restriction enzymes? Because these enzymes were actually found in bacteria. And why do bacteria have these restriction enzymes? Because these bacteria, they can be attacked by viruses. So when the virus attacks, its DNA goes inside. To protect itself inside the bacteria, nature has given these bacteria these restriction enzymes by which they can cleave the DNA of a virus. And when the DNA of virus is cleaved, when the DNA of virus is degraded, that virus cannot infect. So this is a kind of a natural defense mechanism which has been given to bacteria by nature, and scientists, they have exploited these, or exploitation would be a wrong word. They have made a good use of these enzymes. I'm sorry, exploitation is a wrong word. The scientists, they have made use of these restriction enzymes. They have isolated these restriction enzymes from this bacteria, and now we use these restriction enzymes for doing various experiments in the lab. Whenever we need to cut the DNA, and it's very useful, these restriction enzymes are now sold by companies, they are commercially available. So first of all, when you are using enzyme, you need to know what is the recognition sequence of that enzyme and how does it cut. So we will be so basically here is some information. You can see here, this is example of few restriction enzymes. They are called ALU1, CLA1, DDE1, EcoR5, MAL1. These are the names of the restriction enzymes. This is the recognition sequence. Like for example, ALU1. This enzyme, when it has to cut the DNA, it will first of all recognize this sequence. It is going to cut. It is going to recognize this sequence. If this sequence is not there, it is not going to cut the DNA. And what is its cleavage pattern? So it basically breaks the bond between G and to break the bond between G and C. So this is G and this is C. It's going to break the bond here. And on the other strand, so be careful, DNA is double stranded. Okay, so you have to be careful where this enzyme cuts. So it cuts here, its cleavage pattern is between G and C, and on the second other strand also it's cutting between G and C. So when this enzyme cuts here, so the DNA will be cut, so it will create blunt ends. So these kind of ends, because this CT will be on this side, AG will be on this side, TC will be on this side, GA will be on this side. These kind of ends are called blunt ends. Now here is example of another enzyme. This is called CLAV1. Now its recognition sequence is ATCG80. It's going to recognize this sequence. But where it is going to cut? It is going to cut between T and C. So you can see here, this is ATCG80. It's going to make a cut here. Okay, it's going to cut the DNA here. And in the opposite strand, so it cuts between T and C. So you can see here, on the opposite strand, here is T and C, it's going to make a cut here. Now if the DNA, so this is your double stranded DNA, okay, so you can see here, this is A, this is T, this is T, this is A, this is C, G, A and T. So basically when it is going, if it is making a cut here and it is making a cut here, okay, so it's making a cut here, it's making a cut here, it is going to create the sticky ends. So because if it makes cut here, it will be cutting here, and on the other strand it is going to make a cut here. Okay, I think just one second, yeah, I think here it should be, this is C, it should be G. Okay, there's a mistake here, this is G, this is G, this is C, that's fine. So basically the idea is the two strands are complementary to each other. Okay, so if it makes cut here and it makes a cut here, you can see if the DNA is going to be cut here, this will be AT, this will be CG80 on this side, and this is cut here. On the other strand, if it is cutting here, this will be TA on this side and TACC on this side. So you can imagine the after cutting how the DNA is going to be. On the upper strand, I'm just going to show you with this arrow. On the upper strand, it is AT here and the other side is CG80. Okay, and on the opposite strand, it is TA here and it is TACC here. So the kind of the ends the DNA will be having after it is being cut by this enzyme, they're called sticky ends because they are actually like sticky. So you can see that why they're called sticky and why they're called blunt ends. So they have a different cleavage patterns. Okay, so on the basis, so it's very important to understand how an enzyme cuts. Okay, how an enzyme cuts is very important. So on the basis of how an enzyme is going to cut, we can detect if the mutation is there or not. But we need to do one thing more. So if we have the PCR amplified region of DNA, and after that we have cut with the restriction enzymes. Now a question should come to your mind here, why are we cutting the DNA with restriction enzymes? The reason is restriction enzymes cut in a very specific way. Now what problem we are working on? The problem we are working on is detection of a mutation. Now if the DNA is mutated, which means it is going to have a different sequence. If it has a different sequence, the restriction enzymes, they are going to cut the DNA in a different way. So if you have a normal sample of the DNA, normal person, another sample, DNA sample is from the person who is suffering from sickle cell anemia, okay, and they will be having different, And when you cut with restriction enzymes, those DNA samples will be cut in a different way, and that you can detect in a DNA gel. Okay, now I'm going to explain you what is DNA gel. So DNA gel can be used in various situations. So this is one example. So DNA electrophoresis is basically a kind of a apparatus, and you are going to run the DNA gel at your home, because in this Carolina kit everything is given to you, and you will see that in this document. I have posted this also on Mercer Blackboard at 2-3 places. The Carolina, they have made their own videos. They explain it very nicely, how to pour the gel, how to load the samples. There are 2-3 videos. Please watch them before you actually start doing your experiment, and if you have watched those videos, you will have no problem in doing this DNA gel. The only thing is, right now I'm talking to you about sickle cell anemia and normal samples. The DNA gel you are going to run at your home does not have DNA samples, but they have provided you with 5 or 6 dyes. Okay, they are of different colors. So when they run on the gel, that will give you an idea how does DNA runs on the gel. Now, first of all, what is a DNA gel? DNA gel is basically made of a jelly kind of a material which is called agarose. So this agarose basically solidifies at room temperature, and it is very porous inside, and inside there are very tiny pores. Okay, and when you will make the gel, you will insert a comb before you actually start pouring the gel. So you will insert the comb, and then you will add the molten agarose, and then once the agarose solidifies, you can take that comb out. The reason for inserting the comb is to make the wells. Now, why you are making the wells? Because you have multiple samples of DNA. So wells are like different compartments where you can put your where you can just pipette your different samples. So they are like little rooms for your different DNA samples or different compartments, and we call them wells. Okay, so you are going to add your samples to these wells. So in these wells, when you have loaded your samples, how does the DNA runs in the gel? You apply the electric current. Okay, DNA is negatively charged, and when you apply the electric current, DNA moves towards the positive cathode, which is positive electrode, which is a anode, because DNA itself is negatively charged. Because when the DNA is running under the effect of electric current, if the DNA sample has different fragments and they are of different size, they get separated. Now the fragments which are bigger in size, they will be having difficulty in moving through the pores, because this agarose has inside some pores. So the DNA fragments which are bigger in size, they will stay near the wells, and the DNA fragments which are very small in size, they'll move away from the well. So the small fragments, they run faster, and the bigger fragments, the larger fragments, they run slower. So on the basis of under the effect of the electric current, DNA runs, and if it is a mixture of different DNA fragments, they are going to separate and they are going to move with different speed. And on the basis of how different DNA fragments they are running in a gel, you can detect mutations, you can detect various things inside the gel. And also in these DNA gels, we usually load a DNA marker. Now what is a DNA marker? It's a kind of a reference. So DNA markers are usually sold by commercial companies. You can buy from them. So if you have like 10 or 12 wells in a gel, one of the well you can use for loading the DNA marker. So in that well you can load your DNA marker, and when that marker runs with your other die for this experiment. Basically, if you have, like, you have suppose 7-8 samples, DNA samples, you can load your samples in the different wells, and in the last well you can just load the DNA marker. So when the DNA marker runs along with your other DNA samples, it is also going to show you some fragment pattern. But in the marker, we know that what is the size of every band of the DNA we see, okay, because it's a reference. The company tells you if you run it in the gel, you will see 6 fragments. The size of the largest fragment is suppose 10,000 kb, then the second one is 5,000 kb. So basically it's a marker, it's a reference. So the DNA fragments inside the marker, what is their size, we know that, okay. So when we run the marker along with our samples, other samples, we can have an estimate of what is the size of our DNA fragments in our samples, and that is by comparing the DNA fragments from other samples with the DNA fragments which are in the marker. So that is the role of the DNA marker. So basically, after you have done the restriction digestion of the 2 samples, so you will take your DNA sample, you will do the PCR from the mutant, the sickle cell and the normal. You do the PCR, you amplify, then you cut with the restriction enzymes. Now because the DNA sequence in these 2 samples are different, restriction enzymes are going to cut them in a different way and will give different kinds of DNA fragments, and that you are going to detect in a electrophoresis, in a DNA electrophoresis in a gel. But like, if suppose you have 2 samples, one is from normal person, one is from your mutant, after you have done PCR, restriction enzyme digestion, you have the sample, you can load the samples from the normal person in one well. In the second well, you can load the sample from the sickle cell anemia patient, and in the third well, you can load one marker and run them together. So after the gel is run, you are going to see the DNA fragment pattern on the gel, and in the marker, how the DNA fragments they are running, we know what is the size of each band in the marker, and we can compare the DNA fragment pattern in the normal sample and the sickle cell anemia by comparing it with the marker fragments, and we can have an estimate of the how much is the size of each fragment in those 2 lanes. So that is what is done in electrophoresis. So what you are going to do, you are going to do this electrophoresis, okay. So basically this is the material which is in your kit. So you should check if you have everything. So this is a comb, this is a carbon fiber sheet, this is the black electrode, this is red, this is TBE buffer, you will need this buffer. These are all different kinds of dyes. This is syringe, these are tips, this is tubing to make the adapters, this is the agarose given to you here, and this you need to use the microwave to, first of all you need to warm it, and once it's warmed and it's then you can pour it in this kind of the electrophoresis chamber. So all the information is very clear. So basically this is the material. The very first thing you're going to do is you're going to pour the gel, okay. So this is the chamber and this is the comb in which you're going to pour the gel. So watch this video. This video is provided by Carolina, and they explain very nicely how to pour the gel, okay. So watch this, you will understand how to pour the gel, and then after that there is an activity, and out of this activity I have made your worksheet, okay. So that is worksheet number 1, which is on Blackboard in the week 7 folder. It is going to be graded, it is worth 15 points. So it's basically this activity I have made your lab worksheet, okay. And in that worksheet, after this activity is done, because this will be done on paper, these sequences are given to you. This is wild type hemoglobin, this is mutant, and also here you need to use this restriction enzyme, time, how does it cut and everything, it is provided to you already in this worksheet. It's, if you go back, there they have given us a lot of given us information about 5-6 restriction enzymes here. So you will be using this one. So I have provided that information in the worksheet also. Okay, so for this activity you can do the worksheet, and also in the worksheet I have added one more question which says that paste the picture of the gel. So after you are done with your second activity, so your worksheet is basically activity number 1, and I also need the picture of the gel which you have done at home. You just need to paste the picture. So once your gel is done, take the picture and paste that picture of your gel in your worksheet. Okay, so so basically these sequences are given to you, and they are asking you to use this restriction enzyme, and you know how they cut. So on the piece of paper you can try how this restriction enzyme is going to cut this DNA, how it is going to cut this DNA, and then on the basis of that you can fill the information. You can just just try on a piece of paper how this enzyme is going to cut this DNA, how it's going to cut this DNA, and when you have done the restriction enzyme digestion, after that the next step is to do the DNA electrophoresis. So on the basis of your results, just write down here where the DNA fragments will be visible. Okay, so so this is the same, there's only one enzyme. It can cut this also, it can cut this also, but how does it cut this and how does it cut this will be different, because the sequence of DNA is different. So you need to what I would suggest, take a piece of paper, write down this sequence, write down this sequence. That table should be in front of you, or you, if you know what is the cleavage pattern of this enzyme, then just make the cuts. Okay, just make the cuts and then write down how the DNA is going to, how that resultant DNA will look like, or how is the DNA after the digestion. Are there blunt ends, are there sticky ends? So just make a picture of that. Okay, and after you have made a piece of this picture on a piece of paper, you will be able to tell us on this gel where your DNA fragment bands are going to be present on this DNA gel. So the size of the DNA base pairs is also given. This is 15, this is 10, this is 5, and this is 1. And this is, they have given you one sample already. This is unknown beta globulin. Okay, basically another thing I would like to tell, now this is wild type sequence, this is homozygous mutant sequence, and in the gel they have a place for heterozygous also. You can just skip this because we don't have the sequence for this. So you have the sequence for this, you have the sequence for this one, just skip it. So basically take these sequences, cut with this enzyme on a piece of paper, and see how the DNA is going to be after it is cut with these enzymes, and how does in the gel that where that DNA fragments are going to run. Okay, so basically these are the wells. If the DNA fragments are heavy, they will be more towards the well. If the DNA fragments are lighter, they can move away from the well. So you can see here it's already shown, this is 15 base pairs, this is 10, this is 5, and this is 1. So 15 base pairs out of all these 4 is the heaviest, so it's more towards the well. And this is 10, then followed by 5, and then followed by 10, followed by 1. So this is your worksheet. Okay, coming back to your activity number 2, then you need to you need to prepare the syringe, because with the help of the syringe you are going to load your sample. So watch these videos. I think this video and this video is actually same. Watch these videos, they explain very nicely how to make this syringe and how to load your samples. So you have to load all the dyes which have been given to you in the kit, and then you have to circuit. Okay, then you have to make the circuit. You need to use some batteries. The information is given. What kind of batteries? I think batteries are not provided, you need to buy them. So you will buy the batteries, you will connect the batteries, then black one is negative, red one is positive. Just make the connection, and once the connection is made, the gel will run, the DNA will start moving. So once the fastest moving die has reached up to here, you can just break this circuit, and all your samples, all the dies, they will be running on the gel. After you have disconnected it, you can take the picture of the gel with all the different dies which have run on your gel, and you can take the picture, then you can paste this picture in your worksheet. So this is this lab. So I hope it is clear, and if you guys have any question, you can let me know. Thank you very much.